Now we shall discuss about the biochemical factors that are associated with the innate immunity. First one is secretions of the skin. Sweat is a clear salty liquid produced by the glands in the skin. High concentration of salts in the sweat has microbicidal activity. In addition, sweat is also slightly acidic, and this acidity also has microbicidal activity. The secretion of the sebaceous and sweat glands serve as antiseptics as they contain lactic acids and fatty acids, which has bactericidal and fungicidal activity. But certain areas of our body, such as soles of the feet, are deficient of sebaceous glands. Because of this, these areas are easily attacked by fungal growth. Hence, these areas are known as alkaline gaps. Second factor is secretions of the digestive tract. The high acidity of stomach also has microbicidal activity, and this is due to the secretion of hydrochloric acid by the oxyntic cells that line the stomach. Third factor is human milk, which is rich in antimicrobial substances such as lactoferritin and neuraminic acid, which fight against E. coli and staphylococci. And next one is nasal secretion and saliva, which contain mucopolysaccharides that inactivate certain viruses. Next biochemical factor is lysozyme, which is also known as N-acetylmuramidase. Lysozyme is present in tear, nasal secretion, saliva, polymorphonuclear leukocytes, human milk and other tissue fluids except cerebrospinal fluid and urine. Lysozyme break down peptidoglycan. Peptidoglycan, also known as murine, is a major component of gram-positive bacterial cell wall. By breaking down peptidoglycan, lysozyme compromise the integrity of bacterial cell wall. As a result, bacteria are lysed. In the presence of additional serum complement factors, lysozyme also inactivates certain gram-negative bacteria. Next component is interferons. Interferons are signaling proteins produced by cells in response to the presence of viruses. They are antiviral agents which inhibit intracellular viral replication inside the cells that are infected with viruses. These proteins inhibit viral mRNA transcription, thereby disrupting viral life cycle. Interferons also increase the activity of natural killer cells, thereby providing immunity to cancer. Because of anti-cancer and antiviral properties, interferons are used in the treatment of viral infection and also in cancer chemotherapy. Large amount of interferons are produced by recombinant DNA technology, in which the gene encoding interferon is introduced into E. coli, where interferons are produced. Commercial production of interferons by recombinant DNA technology is a major breakthrough in science. Complement system is another biochemical component of innate immunity. Complement is a complex system of enzymatic proteins found in serum. They are activated by antigen-antibody complexes. Activated complement components increase phagocytosis of pathogens and thereby their destruction. Complement system operate by three pathways. They are classical pathway, alternative pathway, and the lectin pathway. The main function of the complement system is the opsonization of microorganisms and formation of membranous complex which cause lysis of the pathogens. Next factor is properdin. It is a plasma glycoprotein encoded by CFP gene. Properdin along with complement and magnesium ion cause the lysis of gram-negative bacteria. It also inactivates certain viruses. Next factor is secretions of bacterial flora. Bacterial flora of the skin produce antimicrobial substances such as bacteriocins and acids. In addition, bacteria live in the human gut as commensals. These microorganisms also produce antibacterial substances such as colicins and acids, which prevent the entry of other microorganisms into the body. When these microorganisms are removed by antibiotics, pathogens get an easy access. In addition, lactobacilli of the vagina also produce antimicrobial acids, and finally, the mucous membrane also secretes antimicrobial substances which inhibit the proliferation of pathogens. Next factor is semen, which also contain various components such as spermine and zinc, and finally acute phase proteins, which are a class of plasma proteins whose concentration either increase or decrease in response to inflammation. One example is C-reactive protein, which binds the C protein of pneumococci. The concentration of CRP increase in response to inflammation. It acts as opsonins. Bacteria-bound CRP activate the complement system, thereby facilitating their uptake by phagocytosis. So friends, this is all about various biochemical factors associated with innate immunity. For more updates, please subscribe my YouTube channel, Sam Biology Class.